Hi, everyone. Thanks so much for joining us for How She Does It. On this show, we talk about all things women, money, and power. I'm Karen Feinerman. We are nearing our 50th episode of the How She Does It podcast, which I can't even believe. And I have met so many women doing extraordinary things, and I wish we were at the point that it wasn't unremarkable that they were women. But unfortunately, we're not there yet. So nowhere is this more important than in the field of STEM. Science, technology, engineering, and mathematics. And the good news is that more women are choosing career paths in the field. We haven't looked at women in the field of science yet. And what an unbelievably good place to start is with my guest today, Paola Arlotta. Paolo is a neurobiologist and stem cell scientist at Harvard University. Her work centers on understanding the human brain, how it's generated, how it functions, and how it's affected by neurological diseases such as autism spectrum disorder, and schizophrenia, which she will explain to us in an understandable way. I promised. I asked her to explain it as if I were a golden retriever. We're going to get into her work exploring questions in neurobiology that have never before been accessible to scientists and the obstacles that she's worked to overcome as a female scientist. But before we get into the episode, I want to make a quick announcement. Next week will be our final episode of season 1. We'll be back later this year, and I can't wait to see you then. Paola, welcome to the show.
Thank you, Karen. Pleasure to be here.
Okay, so a little bit of background. I know you grew up in a very small village in Italy. And how did you decide that winemaking, literally, which I think is what your family did, was not for you?
It was a tough one. I must admit, I still kept that one little vineyard that my grandfather had in case in the future winemaking is on the horizon. I feel that in a way, I always was a scientist, at least in terms of being curious about the world around us, how things really work in life, in nature. Spend a lot of time outdoors looking at how insects and worms and things like that interact with the world. And got fascinated about this whole thing. Nobody in my family was a scientist, so I didn't have, I guess, that formal introduction to science. But I was lucky to have amazing professors, especially in high school, that inspired me. And the rest sort of came together a way or another. And I'm privileged that I could become a scientist and continue to ask about how the world around us works.
It's such a wonderful story to hear that there was some teacher along the way who really changed the course of some people's lives. I love those stories. So that happened for you. Okay.
Oh, so, it did happen for me, for sure. I wouldn't be here.
So, and here is a very incredible place to be. I have to jump right to the sort of most, I guess, I don't know if it's the most famous thing about you as a scientist, but tell us about brain organoids, which I had never heard of before. What are they and how have they, this is your life's work. What is it all about?
It sounds pretty crazy. It will sound pretty crazy, but what human brain organoids are tiny, tiny little pieces of tissue, of brain tissue, except instead of being made by nature, they're made in a lab, starting from a simple sample of blood that we can take from any one of us. Then that blood is turned by scientists in the lab into a cell called a stem cell. And the stem cells can generate any cell of our body. They're very similar to the stem cells that very early on, when you and I were embryos, very, very a long time ago, began forming all of the cells of our body in the embryo. Now we can make these cells in the lab. And then you can coach these cells to undergo a process of so nervous system development. And slowly over time, the stem cells will form cells of the brain. They'll form multiple different types of cells of the brain in the same order as they're made in the embryo. And by the end of this process, you end up with a piece of tissue. It's about apple seed size tissue, about 5 millimeters in diameter. You can see them with your eyes. They grow in culture dishes or culture flasks in a room called tissue culture rooms in labs. And the amazing thing about these organoids is that they are the very first opportunity for science to watch a process of development or of formation of the human brain. And you may say, why haven't you looked at it before? Like, why didn't you look at the actual human brain? As you know, our brain is made in utero as we develop in the womb. And then even when we're born, it's not finished. It's going to take 20 years or so to finish that brain. I do have a teenager at home. That brain is not finished yet by age 15. And so, especially what happens in utero, we don't have access to. We can't look at it. We can't experiment with, of course, and so on and so forth. And so we don't know very much about how our human brain is made. And as a consequence, we also don't know very much about how neurodevelopmental disease, so a disease of development of the brain, like autism spectrum disorder, how does it work? Where does it start? And so when you said sort of looking at science that was never accessible before, this is what these human brain organoids do. They allow us to rewind the time, start from a sample of blood of an adult person, and watch the development of that brain that happened a long time ago in the womb when that person was an embryo, and understand how it's made and how it's affected by disease.
Is it you always take the stem cell from an adult? And is that because the brain is fully formed? What is the reason for that?
No, you start with a sample of blood. And so you can take a sample of blood from any one of us, including, it doesn't have to be an adult, it could also be a child. And then because the cells in our blood contain the genome of the person, so inside the nucleus, inside the cells, it's our DNA, which defines me as Paola, you as Karen. And therefore, if I start from the sample of blood, I turn it into a stem cell, and then I turn it into a human brain organoid. Every cell in that piece of brain that I'm making will have my genome if I started from my blood, or your genome if you started from your blood. And so you're going to watch how your genome is building your brain. You're going to watch how your genome may actually cause abnormalities also of brain development and function.
Okay, so you created this brain organoid process. Is that right?
Yeah,
it's what does that feel like?
What is it?
I mean,
it's
that's
amazing.
Extraordinary.
It's amazing. It's sort of it reminds you of why we went into science in the first place. I can tell you a little story so I always say we can make these human brain organoids, that are a replica of what you would see in the tissue. But I've been looking at brain tissue, at the actual brain tissue, for a long time in my life. And I know what's inside a piece of brain. There are these cells and synapses, which are the points of contact among the cells, and so on and so forth. They have specific shapes. And so we grew these organoids, not really knowing what we would get inside them. And then we look at them. And the first time that we look at them, we were shocked that a lot of the same characteristics and features that one finds in an actual sample of brain was there too. And it told us that we could use them to understand where we come from with this tissue. Number one, again, it's an organ that really defines us as individuals, as a species. And also understand for the first time, how do we get this pathology that are uniquely human and really sort of pathologies of our genome to a large extent.
All right, so I know you're working on this for years. Just tell me about that moment where you realized, all right, what we have been trying to do, we have accomplished. Like, do you literally say like eureka in a lab?
There were several eureka moments. When we first started this and when this field really started, and of course it's not just my lab, it's other labs now as well, we didn't know if this could really happen. It's one thing to ask a stem cell to make maybe one cell of the brain, or to make a structure that from the outside, maybe there are brain cells in there. And it's another thing to actually very reproducibly and consistently produce this structure and looking inside it and realizing that you can walk the same kind of steps of development of the brain that the embryo did when normally our brain is built in the womb, and that nobody had seen that before. So most of what we do as scientists in the lab, this may may come as a surprise, but it's really true. 95% of what we do fails, like in the lab, right? You go in, you do an experiment, you sort of expect failure and you expect to learn from failure to eventually succeed. And so we certainly expected a lot of failure here, but in reality, we were successful more than we expected to be. And we could really have a lot of those eureka moments where we said, wow, we have this. Now we can really fulfill the promise of what this tissue can really allow science in general to do and society to do with it as well.
Okay, so tell us that next step. What is the promise of it? As you can see what's happening in the womb, as the brain is developing, what is it that you're looking for?
If I asked you or anyone, any scientist also, what cell types of the thousands of different types that are present in the brain are affected in autism spectrum disorder, nobody could tell you what they are. If I ask you, when does autism start in the womb? Also, nobody could tell you, and leave alone, what are the mechanisms of this? What are the genes that are involved, and so on and so forth. And so I'm going to take autism as an example, but the same reasoning can apply to a lot of other disorders. And in part, we couldn't answer all these questions because the human brain is a very specialized tissue. So if I compared my liver to the liver of a mouse, yes, it would be different, but honestly, quite similar. But if I compare my brain to the brain of a mouse, it's different. And mice, of course, are the classic organisms that are used to study things in labs. There is a lot that we can learn from mice, but there will be a point where it's so specialized that we cannot really deduct enough. The other thing that happened over the past 10 years or so is that scientists began to sequence the DNA of a lot of patients with autism, as well as schizophrenia and bipolar disorder and so on and so forth. And what they saw is that the majority of these patients have these very complex genetic states. They're called polygenic, many genes and many parts of the genome. They are not diseases of like one gene, one mutation that would give you a disease that you can take the same gene and mutate it in a mouse necessarily. It's more often a disease of many, many sort of positions in the genome that give you a certain probability for developing this disease. And so what scientists wanted to do, and that's what I want to do now, is to take really literally the information from the genome of patients and let that information tell me what may be going wrong or may have gone wrong in their brain as it was developing, or later on as schizophrenia may start. And you say, why do you need a human model for that? You need a human model because I told you can take this sample of blood, and every cell in the sample of blood, I remind you, has the genome of the patient or the person I took it from. So all of a sudden, you can make a brain organoid from the very cells with the very genome of the patient that you're trying to understand. And you can watch this movie and characterize it and understand the process that may have led to some of the abnormalities of brain function or differences in brain functions that we see in the patients. So number one, we're gonna understand and investigate in the actual human brain, carrying the genome of the actual patient we're trying to understand and study and cure, what is wrong. And once we understand what is wrong, that's step number one, to understand what are we going to do about it. Mm-hmm. And so the second part of this, once we understand what might be the mechanisms and the processes from the genetic information that may affect the brain in these patients, then the organoids can also become, and they're already becoming, a platform, meaning a piece of tissue, right, on which you can begin to test drugs and treatments to see how the cells of that very patient would react to them.
And so you have millions of cells of that same patient, so you can test various things on the same patient at the same time.
Absolutely. And in fact, you can do more. So each and every one of these organoids contains millions of cells of the brain. We know from our work and that of many others that these cells resemble the ones that you see in an actual human brain. They're still not brain, they're organoids, they're replicas, right? But you can experiment with them in ways that you can't do with the actual brain, which happens to be inside our head, and we can't take a sample of of brain, but we can take a sample of blood. So many cells, millions of cells that you can study, but also we figured out very recently that we can even grow these organoids starting from the cells of many people. So now not only you have in a single organoid millions of cells of the brain, but you have millions of cells of the brain from many different people. And now you can ask questions such as, how would the cells that came from Paola react to this drug? And how are the cells that came from Karen reacting to this drug? And now we can understand a little bit better whether I should be in a clinical trial testing that drug or whether you should be in that clinical trial testing that drug. I think we can really transform the way we think about the development of therapeutics, even at this fundamental level.
Okay, so that seems like it skips tons of steps of testing on the wrong group or testing on too wide of a group. So I would imagine the hope for drug development is, I mean, is this something you think you can cure, schizophrenia?
That's certainly the hope. I first would like to understand it. Like I would really like to understand, so the foundation and the mechanism of autism spectrum disorder, how does it come about? And similarly for schizophrenia. Then I'd like to understand whether human tissue, human brain organoid tissue that comes from different patients could actually be the tissue on which we develop even the drugs. And I would like to understand whether organoids can be used as avatars of our own brain, which we cannot test for all the reasons because the brain is not accessible. Whether organoids can become the sort of the personal avatar to understand how the cells of our brain would react to therapeutics and drugs before we give them to the patient. Eventually, my prediction is that we're going to get so good at this that we are going to be able to predict the likelihood by which different people may respond to new drugs even before we design and effect the clinical trial. So the stratification of patients based on the likelihood by which they will respond to the new drugs, it's something very exciting, and it's something that may actually help us get to a better place for understanding why many of the clinical trials for drugs that should affect the brain that worked nicely in mice actually fail the moment they're tested on humans. Maybe we had the wrong group of humans, and also maybe humans are not mice when it comes to our brain.
So could you also test for dementia or Alzheimer's or?
Yeah, the sort of the promise of human brain organoids is that of being able to model, so to allow a scientist to look and study a piece of tissue that would have processes in it happening that resemble the human condition. And there are many, many different pathologies, neurological diseases of the human brain, and they are not equally easy to model in an organoid. There is a reason why we're starting with the neurodevelopmental and neuropsychiatric. Neurodevelopmental disorder, much like this tissue, as a developing tissue, it's young in a way, but it's getting better and better at modeling also aspects of brain function that happens later in our lives. And it will not take too long to be able to model more and more aspects also of late onset diseases like Alzheimer's disease and Parkinson's disease, for example. A lot of work has already been done. More will happen. It's just a matter of time, I think.
So right now, what you're seeing is what one would see in the womb if you could. Is it in real time? Does it change over 9 months dramatically?
Oh, good question. Very good question. This is a fundamental question, Karen, in biology. I'm happy that you're asking it.
All right, I got one.
It takes time. It actually takes a long time to turn that stem cell that we made from the sample of blood. That step doesn't take very much time, blood to stem cell. But then stem cell to complete brain organoid, it's months and months, in the order of like 6, 7, 8, 9 months to just make the cells of the brain. And that's because we develop really slowly. It takes a long time for our brain in the womb to be made, and it takes also a long time. It's the speed of development of the species. Mice go faster, human go slow, and that happens in vitro as well, so in the organoids. It takes a long time, but it hits some, although not all, of course. It still is a replica. We shouldn't call these organoids brains. They are replicas of the brain that can be studied, made in large numbers in the lab, so that we can actually test treatment, genetic states, interventions on an actual model of the human brain. It's really a model.
Okay, so but it's not a sentient organoid.
Absolutely not. Absolutely not. So, but you raise a very important topic of conversation here. If I had told you, look, I take a sample of blood and I make human liver, you would have said, whatever.
Cool. I would say cool.
Cool, maybe.
Yeah.
Exactly. This is no offense to anybody who studies the liver. It's a pretty cool organ that we need. But if I tell you I'm taking a sample of blood and I'm making human brain, or human brain organoids, I should say, the reaction is different. And that's because the brain defines us. It's a unique organ in our body. It's very specialized. It makes us do the kind of things that only human beings can do. This conversation with you today. And therefore, this work is not easy work from an ethical perspective. It really has to happen within the right ethical framework. I've invested almost the same amount of time as I invest in science, almost really, in engaging in ethical discussions, not just with scientists, but with society in general, and most recently with the National Academy of Sciences, really figuring out where are the boundaries of this science, what are the questions that we should be asking about what's inside these organoids and what these organoids can do. As of today, I'm pretty confident, as I say, that they are really replicas of the tissue. They don't have the complexity, not even close, of what our brain has, and that they are not conscious or anything like that. This said, I think that this is science that moves fast. It's very powerful, could impact society really beautifully, but we need to also think about the ethical implication and constantly ask ourselves those questions. So progress the science, but make it move the right way.
Mm-hmm. So let me ask you, the way you sort of lead as a woman, how did you decide what kind of leader you were going to be, and your being a woman, how relevant is that to what kind of leader that you are?
It's very relevant, and you're really asking a question that gets at the core of it. I think it's very important to remember that, yes, there are many ways of being successful, and everybody needs to find their own way. But for me, it's also important to remember that I want to do this while also staying a woman, dressing like a woman, talking like a woman, and acting like one. I think this is very important because every aspect of your identity sort of then has to be part of who you are as a leader. And I'm going to tell you maybe an embarrassing story, somewhat embarrassing. I was an assistant professor many years ago, and I was sort of my first round of major talks and lectures in front of big audiences. And a very good friend of mine who was a professor at Oxford, also in the science, he was in the audience. And I go up there and I give my talk and I calm down, and she leans over and says, "What's wrong with your clothes?" And I'm like, oh my God. And so all of a sudden I thought maybe there is something on my shirt, or so I look at myself, I said, "What's wrong with my clothes?" And she says, "Do you go out with your clothes as well, like for dinner?" I'm like, absolutely not. This is sort of like my uniform, right, to give my talk. And I realized that I had this super boring, sort of monastic looking clothes because I wanted to be in between invisible and maybe looking professional, but the professional made me look a little bit like a man.
Right. Well, you wanted to get rid of any hint of womanhood.
Of like, yes, being neutral. And with time, I realized that actually that's a mistake, at least for me. You be you, and began to act differently, to dress differently, to do the kind of things that me as Paola would do as a woman. I think it's important, or we're going to be negating in a way an aspects of ourselves that it's very real. And it's important to mention.
It's a great story because it sort of allowed you to clarify, that's not me. I'm not going to be doing that. So did you have any role models or any mentors when you first went down this path?
Well, I got inspired to start in science literally by a high school professor who, in this tiny public school, in this tiny town, instead of simply teaching us science, would take us to the lab and teaching us science through experiments. And that is what hooked me, what told me that I loved this, that I had somehow to do it. What I didn't know, didn't realize, that the path was going to be at times, let's say, interesting. It's different today, I have to say, or it's much improved today. So I have great hopes for my daughter's generation that things will be more balanced and there will be more women, not just in science, but in leadership in science. When I was growing up through the ranks from assistant professor to full professor, that was really the time when I realized that there is a lot of difference in the way we do things. And it was very different for me to navigate this as a woman. I imagined that as if I was a man. I'm saying this because before then, as a student or as a postdoc, I never felt that way. So I always felt that the opportunity was equal, that we were all coming from different walks of life. There was no difference. The difference for me was when I started my own lab. It was at a time when everything was happening at the same time. I wanted to have children. I was sure I wanted to have children. I wanted to be a mother, and I had a certain expectation from myself for what kind of mother I wanted to be. And there was always this constant fear that I couldn't be a good scientist or I couldn't be a good mother. It was like one or the other. And it took me a while to realize that all I had to do, or that what I had to do, was to do me, meaning do things the way I thought would be effective and good for me to be successful, and that there are many, many different ways of being successful in science. I had incredible mentors, including many men in my department, which was populated at that time mostly by men. We were 2 women in the department, junior women, but they were extremely supportive of us and our growth. And I had one professor at Harvard Medical School, a woman, Connie Sepko. She's amazing. She studies the retina. And she did give me a lot of advice here and there that allowed me to build the confidence to really say, I'm going to be successful in my own terms. And that meant going against the current a bit about when I could come to the lab in the morning, or how I would do my work, when would I do it, how would I teach my students. And at some point, I realized that the way I was doing it was not just important for me, but it was very important for the many women that I happened to train in my lab. I had a lot of exceptional, and I should not use this word, they were not exceptional, they were great scientists. Like there are men, great scientists that came to my lab, and they were looking at me to see whether this was a path for them or not. So I felt a bit of responsibility that I needed to do it my own way to show that there was perhaps another way to succeed. Fully understand it, I could have failed. But again, scientists fail all the time, so that's okay.
But did you ever think, oh, you know what, I'm gonna take a pause in my career? And you did.
I did think about it. And I think many women think about it. Now, this you have to put in contrast with the incredible determination I had. I tend to be, if I decide to do something, I'm going to do something, right? So there is very little that can dissuade me. If I feel that is what I want to do, I typically do it. I tend to be a bit stubborn that way. Everybody can tell you. So it was not out of lack of interest, lack of determination. I've always thought that this was my path and that this was the correct path. I could not have become anything else but a scientist. So I was lucky that way. So I didn't have all those doubts, right? The doubts that I could become a scientist, the doubts that I had the chops to do it. But it was taking away a lot of other things in my life that were very, very important, especially related to my kids. They were very young. And so there I really owe it to my husband. We were doing everything together and everything. So everybody was picking up a piece of the story here. And I stuck with it at great, I would say, at times, personal cost, meaning that I didn't want to take it away from my kids and I didn't want to take it away from my lab. So there is only one place where you can take it away from, and it's you. So not much free time for hobbies or anything like that, for sure. And long nights at the computer. Yeah.
Is your husband a scientist also?
He's not. He works in an architecture company, and he decided a long time ago that we were going to balance this out so that, we wanted to do different things in life, and he was always there to support me. And I really think that there were times where I don't know if I could have done it. And so without all that support, because it's an all-encompassing job and you are expected to do a lot of things, I think it takes a little longer for women to realize that you can do these things. Definitely you can, and you can do them in a different way. It takes longer to realize it because we don't have enough role models. Count the number of women that are older than you relative to the number of men. Just a fact that you don't have as many, and many have lived a different experience in science than the ones that I lived. And today, my postdocs and students certainly live in a world that is different from the one I grew up. So role models, Karen, are very important. And that's why I think that despite the fact that at times it takes it away from the lab, from science, it's important to have leadership positions to just set the tone for a slightly different way of doing things and lead by example. Yeah.
So leading by example, do the women in your labs, do they feel like, all right, I could do this and have a family?
I sure hope so. I sure hope so. We even started in the lab a baby clothing recycling path whereby these clothes go from the one postdoc to the next, and I was very proud that for a very long period of time, pretty much every postdoc that walked through my lab had a baby at some point. I don't know, but so I hope that by showing that I did it a certain way, that they can take all the space that they need and still be very successful. I'm very proud of where many of these women went after my lab. And in fact, you can say that the most success they got was afterwards when they did their own thing, right? Yeah.
Well, that must be very fulfilling as well. All right, Paola, thank you so much for explaining your incredible work to us in a way that is understandable. And years down the road, where do you think we will be in terms of ability to treat diseases and have drug development really be accelerated by the work like yours?
I'm gonna predict that we're gonna see a leap forward in the next 10 years or so. One of those changes in discovery and understanding in science, that instead of making us progress slowly and surely towards a better way of developing drugs, let's say, or understanding human disease, we're going to leap forward and skip a whole bunch of stuff. And I think this is because we live in a sort of almost like miraculous time when a bunch of critical technologies and critical knowledge have come to bear all at once. We can make, again, from blood and stem cells, human organoids of every tissue in the body. These are avatars to begin to understand how this tissue is made, but also how it responds to therapeutic, how it's affected by disease, and so on and so forth. We did not have that as massively present as it is right now. We have technology to look at all sorts of things about every single cell of our body, from the genes that we express, to the proteins that are there, to how cells react to things. And then, as you know, we live in a world of machine learning and AI that is already impacting completely our life in so many different dimensions, but it's going to have a great impact in science and biology as well, because it would allow us to really take all of this biological data, on which we can now produce, thanks to this technology, large and large amounts, and begin to help us understand what the data is telling us and begin to predict how our own brain or liver or lung will respond to drugs and disease. Maybe one day without even having to do an experiment. And so I like to think that some leap is coming whereby we are going to learn things in a very different way, and we're going to impact society and the way we think about disease and their treatments in a different way, not just incrementally sort of distinct, but very new. And I feel pretty fortunate that I'm going to get to see this, hopefully, in the next
and be
years.
A part of it. And be part of it,
perhaps. Yes.
Yes. Wow. Okay, here we go. This is just would you rather. The only challenge is just say the first thing that comes into your head. Would you rather be working in the lab or writing a research paper?
Working in the lab.
Lessons in chemistry or hidden figures?
Wow, lessons in chemistry.
Okay. The coast of Italy or the Science Center Observatory at Harvard?
The coast of Italy. Who would
Okay.
not go to the coast of Italy? Which coast of Italy? We're talking about the south, right?
Well, you
The
tell me.
Amalfi
Okay.
Coast,
Okay,
yes.
the Amalfi Coast. Okay, just to clarify. Okay, would you rather be in a lab coat or jeans and a t-shirt?
Lab coat,
Would you rather wine or limoncello?
wine.
Would you rather laugh uncontrollably or be moved?
Laugh uncontrollably.
Yes, that's
Yes.
my choice too. Okay, here's a question. What have you changed your mind about in the last year?
About what you can do at any point in your life. I had a very specific sort of built-in view, maybe from my upbringing. I'm not sure where it came from, that there is a specific sequence in which you do your things in life. And then sort of you miss the opportunity if you haven't done it at the right time. And I lost a bit of that determinism for what I can do or what I want to do. I began to do things I probably should have done 30 years ago, such as rock climbing, ice climbing. I learned how to make pottery, which I thought I had zero artistic talent. I still think I have zero artistic talent, but hey, I can make pots. And I think I've always thought that, I don't know, I think I have somehow I have a certain level of creativity and I like to space out and think outside the box and change my mind all the time. But I wouldn't apply that to every sector of my life. And yes, I've been doing it recently.
All right, well, good for you. Okay, last pair of questions. What's the best investment you ever made and the worst investment you ever made? And investment could mean anything.
Best investment. I've invested in family and I've invested in lab. And it's really the combination of those two. So career, as well as what I wanted to do more as a person, as a woman, as an individual. And I didn't compromise at the time when I thought I should. And that was a great investment. And then the worst, one night at about 3 a.m., I bought one of those mini pie maker. Have you seen those? They sell them everywhere.
No.
Anyway, this was 15 years ago in the middle of the night as I was writing a major grant and I just couldn't see the end of it. I bought this thing, it's still in the box in my basement as a testimony to what you shouldn't buy in the middle of the night when you're writing a grant. That's probably a silly one, but bad
But
investment.
you remember it. All right. Well, Paola, thank you so much for spending the time explaining it in a way that everyone can digest it. Thank you.
Thank you, Karen. Thanks so much.